Our project is the structural insights towards replicating an artificial genetic polymer using the polymerase chain reaction.
The polymerase chain reaction, or PCR, is a robust amplification technique that is used widely in research laboratories but also has practical applications in many other fields. Synthetic biologists recognize that PCR technology does not exist for XNA, which are artificial nucleic acids. The XNA analog that we are interested in studying is ribose nucleic acid, or TNA, an analog that has a 4-atom sugar instead of a 5-atom sugar, seen in DNA. TNA has many interesting properties that make it a promising candidate for therapeutic and diagnostic applications. Using an engineered polymerase, we want to investigate the structural determinants of TNA replication. For example, how can we engineer a polymerase for TNA PCR?
The engineered polymerase being used in our experiment, also known as 1092, was developed in this lab for DNA-dependent TNA synthesis, but we also wanted to assess it for TNA replication ability. Templates with 1 or 2 TNA residues, respectively, were chemically synthesized and were used to investigate the 1092's ability to incorporate TNA residues on a growing DNA primer strand, resulting in TNA-TNA base pairs. Following an incubation of the N+1, N+2 samples overnight, the polymerase's activity was confirmed through the use of a denaturing page gel, which indicated that 1092 had the desired TNA replication ability for further crystallographical studies. Moving forward, we will continue with our established protocol for protein expression and purification from E. coli. Once we have obtained pure protein, we are able to set it up for crystallization trials. The resulting crystals will be a polymerase annealed to a primer template duplex in a closed ternary position with a TNTP nucleotide trapped in its active site. We can utilize this crystal structure to determine the chemistry and mechanism of TNA nucleotide addition by 1092 for TNA replication. This information can then be used to optimize protein engineering strategies and make a better polymerase. Thanks to UROP support, we have been able to conduct these preliminary primer extensions, protein expression, and purification. Thank you.